So the few lectures that are coming are the adrenal cortex and its hormones. So the learning outcomes of this session or this lecture, by the end of this session, you should be able to describe the gross anatomy of the adrenal cortex. You should be able to define the different classes of steroid hormones and major sites of synthesis within the adrenal cortex. Appreciate the biosynthesis pathways of steroid hormones and the significance of some of the enzymes. Now, this part is biochemically quite complicated, so you don't need to know everything. It's just to understand a little bit, because as we will see when we touch the disease side tomorrow or the disease after the next lectures, that will be important. Okay, so just to have a little bit of understanding of the biosynthesis pathway. Explain the regulation of the secretion of corticoids, and describe and discuss the mechanism of action and physiological action of corticosteroids. Now, the last two parts here, which are the physiology, we will be covering tomorrow lecture. Today, we're just going to explain the anatomy, and we're just going to be explaining how these steroid hormones are produced. So we're going to start with the lecture outline, the anatomy of the adrenal gland and the adrenal cortex. We're going to move into classification of steroid hormones. We will have a view on the steroid hormones synthesis, structure and biochemical properties of the steroid hormones, regulation of glucocorticoids, secretion and their physiology, and regulation of mineralocorticoids secretion and their physiological actions. Obviously, by the end of this lecture tomorrow, because this is all in one pack, you will understand the pathway that we were talking about, the hypothalamic pituitary adrenal axis, and we're going to make emphasis on the peripheral organs. So the lecture that we did last week will make more sense because you will complete the part of the organ target and the synthesis and the production of these hormones. So remember from yesterday, the adrenal or supraadrenal gland is above the kidney in this pyramidal shape, and it's formed of two components, the adrenal cortex and the adrenal medulla. What does the adrenal medulla secretes? We studied yesterday catecholamines, and is form of nervous system coming from the ganglion and the neural crest. We will rehearse this again. The adrenal cortex comes from another different tissue. So anatomically, just to recap from yesterday, the adrenals lie on top of the kidney, and they are called suprarenal glands in the US. They are divided into the outer cortex, which forms part of 90% of the volume, and the inner adrenal medulla, which is here, which forms part of 10% of the volume. The medulla, as we studied yesterday, is concerned with the stress response using catecholamines, and also the cortex, this part here, is concerned with also stress response sodium and glucose homeostasis in the body. The adrenal cortex, unlike the adrenal medulla, is essential for life. So if there is no adrenal cortex production of steroid hormones, then it leads to death. So it's very important for the homeostasis of the body to produce these adrenal cortex hormones. Again, just to recapitulate, right, how the organ is developed. Remember, there is the adrenal enlarge or the primordium, which comes from the mesoderm, and these cells pack together to form the adrenal enlarge. This adrenal enlarge will be invaded or introduced by cells from the nervous system in the neural tube, right? Some of them are precursors of Schwann cells and some are neural crest cells. So what happens is that they migrate, they enter the primordium of the adrenal gland, and they form part of the medulla inside. So the medulla has nervous system origin, sympathetic, and the medulla or the cortex is formed from the mesoderm cells here. So let's go a little bit more in detail, because today, I'm afraid, my friends, today things are getting a little bit more complicated, right? It's not an easy organ, okay? Very small, but it produces lots of different hormone synthesis, right? So we have here the adrenal gland, which is above the kidney, that has this pyramidal shape. Inside has the medulla and outside the cortex. If we do a cross section of the adrenal gland, what we have is this area. There is a cartoon in A and a histological section stained with hematoxylin and eosin. This area, right, of the adrenal cortex can be subdivided into different cell types and structures. So the outermost layer is called the capsule, and the second layer is called the zona glomerulosa. Lower down, if you look at the capsule, is the outermost layer. Then there is a small cells of zona glomerulosa, and then zona fasciculata. What you can see is that it's formed of these fibers which are elongated. Also of zona reticularis, which form a reticulum. And just below, there are the cells packed that form the adrenal medulla. That's what we studied yesterday. If we look through a histological section of H and E, we can see here the very thin layer of capsular cells. And also we can see the zona glomerulosa. And after the zona fasciculata and the zona reticularis. And what we can see here is the adrenal medulla, which we studied yesterday. But as you can see, they are in intimate contact. What happens if we look here, a cross section of adrenal gland in adult? We can see the fibrous capsule of the capsule cells, again zona glomerulosa, zona fasciculata, and zona reticularis and the medulla of the adrenal gland with the central adrenal medulla vein. Why is important these different zones or different zonations? As you will see through the talk, each zona produces different enzymes that will modify the steroid hormone to become corticosteroid or sexual androgens or different hormones by modifying the initial component, which is called cholesterol. So the zona glomerulosa makes up to 5 to 10% of the cortex. Cells are small and round, as you can see here, like here on the top, and have lipid droplets, but very few. The zona fasciculata on the other side makes up 60 to 75% of the entire cortex. Cells are large and contain large number of lipid droplets. And why lipid droplets? As we are going to study, steroid hormones are lipidic or hydrophobic, right? Comparing to the normal other hormones, which are hydrophilic, these ones are hydrophobic, right? The zona reticularis makes up to 20-30% of the cortex. Cells are intermediate in size and contain few lipid droplets. So here is a little bit more of the histological anatomy. We go from inside to outside, or from outside to inside, doesn't matter. Capsula, zona glomerulosa, zona fasciculata, zona reticularis, and the internal medulla. So we can look here at higher magnification, how you can see it in the microscope. The zona glomerulosa, you can see glomeruli form. Fasciculata, remember, it forms columns like this of cells arranged in columns. And then we have the zona reticularis. So we have done the major anatomy of the adrenal gland and specifically of the adrenal cortex. But what we need to do is to classify the classification of the steroid hormones, which are the ones that it produces. So the classes of steroid hormones are classified into 5 classes of steroids and 2 families: the adrenocortical steroids and the gonadal steroids. So in one hand we have glucocorticoids and mineralocorticoids that are produced by the adrenal cortex, and also we have androgens with estrogen and progesterones, which are formed by the gonadals. Okay, and as we will see, this will be also secreted and modified from a unique type of hormone, but we will touch on this later on. And each one has specific functions in the body depending whether the receptors are there in the target organs. So corticosteroids, adrenocortical steroids, are steroid hormones synthesized and secreted by the adrenal cortex. So what are steroid hormones compared to the other hormones that we have studied in the past? the main component of the steroid hormones is that it starts as cholesterol. Cholesterol can come from the liver or from our diet, but also is present in the cell membranes. And this is cholesterol structure that has this defined molecular structure, which we can see here in A, B, C, D. And you're gonna see that the different enzymes, what they do is that they modify this structure, right? And add carboxyl group or modifying groups. So this structure is a tetracyclic skeleton or cyclopentane phenanthrin. You can see here, right, the classical structure of cholesterol, and all the hormones that produces the adrenal gland derive from modification of this initial cholesterol. Important thing to remember is that the cholesterol, right, compared to the other hormones that we have studied, the other hormones are hydrophilic, so they can travel in the blood very nicely and freely, right, to their target organs. Cholesterol is hydrophobic, so doesn't like water, and therefore, because it's a lipid component, can go through the membrane of the cell without a receptor, right? So the receptors of steroid hormones are found inside of the cell, in the cytoplasm or in the nucleus, but not in the membrane. So the steroid geometry, you don't need to know this by heart, please. You just need to know, for those ones that have an interest, is that it has a special tetraedric, as we said before, right, composition. We're not going to go into the further biochemic of what is weak or not, but basically is a cyclopentane. And this one has the structure of the different carbons, and what you can see, it's got 20 carbon structure. So where are these hormones produced? And adrenocortical steroids, such as, obviously regulate many physiological functions, but mainly to salt, sugar, and sex. So, as we said before, the adrenal gland secretes, here we can see the different zona, capsula, zona glomerulosa, zona fasciculata, zona reticularis, medulla. If we do a cross section of this capsule, we have the capsular artery that provides the blood, and it drains into the zona glomerulosa. The zona glomerulosa will produce mineralocorticoids known as aldosterone. Zona fasciculata will produce glucocorticoids or cortisol, very important for the response to stress. And the zona reticularis, androgens, important in the sexual maturation of the reproductive organs. And then we have the medulla, which we studied yesterday, which has the epinephrine and norepinephrine, right? And it works as paraganglionic sympathetic terminal. Now, each specific cell, like in blue or here of, the zona fasciculata in lilac and the one in green, contains different enzymes which will allow the transformation of this cholesterol into different hormones derived from cholesterol, such as aldosterone, cortisol, and androgens. So corticosteroids are produced by the adrenal, and there are also called mineralocorticoids and are important through aldosterone with the salt balance. Okay, so very, very important. Glucocorticoids will produce sugar and have a diabetic function in the production of sugar from the tissues. And androgens are important for sex and development of the sexual organs. So very, very, very important, and sexual features. Let's start the gonadal steroids. The gonadal or sex steroids are progestogens, also called as progesterone, estrogen, estradiol, and androgens, testosterone. The estrogen confers feminine aspects and affects more the reproductive female function. Androgens give masculine traits, and obviously it more involved in the testes. Now we will study a specific role of progestogens, estrogens, and androgens at the end of the endocrine module, that we have a whole session on reproductive biology. We will study the formation of the gonads. We will study the effect of each of these hormones in the reproductive hormones. So let's look into an overview of steroid hormone synthesis. What are the main differences between the synthesis of peptide and steroid hormones? So let's look into a cell, okay? In one side, you have a normal peptide or hormone producing cell. We have the mRNA that obviously will produce a protein, and this is in the rough endoplasmatic reticulum, or ER, which will maturate the protein, will form vesicles, and then in the Golgi complex will finish this protein complex, and then this will travel to the membrane and being secreted. It also can be secreted through lysosomes, and we have big mitochondria that obviously are important in the energy metabolism of the cells. How about a steroidogenic cell? Steroidogenic cells need to accumulate steroidogenic hormones in form of lipids, and these lipids are formed in droplets. We will see that the mitochondria contains certain enzymes which are called cytochrome oxidases, which modify the lipid to convert it into a different steroid hormone. Then this is further kept, right, into, well, the smooth endoplasmatic reticulum and in big droplets, which then can be secreted through lysosomes. Okay? So those are differences between the cells of the normal peptide or peptide secreting cells and steroidogenic cells. And this is a picture of an electron microscopy picture of endocrine steroidogenic cells. And what you can see very importantly, right? So when you see one of these cells in the microscope, you can see big mitochondria with this shape associated to the lipids or droplets, right? This type of cell, you don't see it in a normal peptide producing cell because the function of the endocrine steroidogenic is to produce steroid hormones which are based from cholesterol or lipid. How about the steroid hormone synthesis, right? And here is where it gets a little complicated. But you guys do not need to know all the pathways because they are complicated. Just have a rough idea. When we come to the disease in the other side, yes, we will study the enzyme that produced the disease. But at this stage, you just need to know a little bit overall. So cholesterol has 27 carbons, right? So all the steroid hormones derive from cholesterol. Remember that the cholesterol comes from different sources, okay? But the liver and diet. Then there are a series of enzymatic steps in the mitochondria and the endoplasmatic reticulum, which are called steroidogenic tissues, that convert the cholesterol into all other steroid hormones, intermediates. Okay? And this normally occurs by taking out carbon groups. We don't want it. We want to modify it. How do we do this? Using decarboxylases enzymes. Okay? Remember, hydroxylases add decarboxylases, and each enzyme will do something different. So we start with cholesterol, then it will take 6 carbons away, and we will end up with progestogens. Okay? Then another enzyme will take 2 carbons and will form androgens, and then 1 carbon more and will form estrogen. Okay? This is to form the sexual gonadal steroids, but also remember that there is the cortisol, right? And mineraloglucorticoids, right? And this basically is what happens to it. So are the modifications, and we will study the enzyme. Don't worry too much about it, but basically there is one branch that forms estrogen and androgen, and another branch that modifies the cholesterol to form mineraloglucocorticoids, which have different functions. Remember that each one of these hormones is produced in one area of the adrenal cortex, in the glomerulosa, fasciculata, or reticularis. So let's look into the sources of cholesterol in the body. So they can be synthesized from acetate to free cholesterol, and this happens in the ER, and this is called de novo synthesis from acetate. Also, plasma membrane cholesterol. Remember that plasma membrane can have cholesterol. Also, intracellular cholesterol esters also present. Cholesterol oleate in lipid droplets found in the steroidogenic cells. That's how it can be accumulated before being modified. And then plasma lipoproteins. And we have different types of lipoproteins. I don't know if you have studied this already in the biochemistry module, but there are low density LDL, which basically travel through the ApoB100 receptor. So there are a lot of ways to obtain cholesterol for the synthesis of the steroid hormones. Sorry, there is also high density lipoprotein, so the low LDL and the HDL. So there are several sources of cholesterol in our organism. Now this is a video to explain you the obtention of cholesterol in the circulation blood. I hope it works. Let me see.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated.
Let's start it again. Sorry. Please pay attention.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis.
Why is this not working?
Instead of circulating freely, cholesterol molecules are derivatized and packed inside low-density lipoprotein particles, or LDLs. A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized by LDL receptors on the surface of cells. An adapter molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane to bend and invaginate, forming a vesicle that buds off inside the cell, taking with it LDL receptors and the LDL particles bound to them. Once inside the cell, the vesicle uncoats and fuses with the endosome, the intracellular compartment that first receives all endocytosed material. The endosome has low internal pH, which causes the LDL receptors to release their cargo. Empty LDL receptors are recycled to the plasma membrane in vesicles that bud off from the endosome. Each LDL receptor makes a round trip from the plasma membrane to the endosome and back every 10 minutes. Meanwhile, the LDL particles need to be disassembled. The endosomal content is delivered to a lysosome, which contains hydrolytic enzymes that can digest the particles. Free cholesterol is liberated, together with amino acids and small peptides generated by digestion of LDL proteins. The cholesterol is then released into the cytosol to be used in the synthesis of new membranes.
Okay, I hope that this was useful. So you understand now how cholesterol enters in the cells and how this is uptaken. Now, this is very important because these hormones do not function as the hormones that you previously seen because they are hydrophobic. Okay? Now, in the biosynthesis of steroids, the cholesterol that has arrived in the cell has a rate limiting step in the synthesis of steroid hormones is in the conversion of pregnenolone, as we will see, which is the first step of modification of the cholesterol. So the cholesterol goes to pregnenolone, and as you can see goes from a carbon 27 to carbon 21. So basically it will take out all this region here, okay? We can see the modification of this tail, right? The rest of the structure is the same. Now, you don't need to know for the purpose of the endocrine the full biochemistry of how this happened. But there is an important enzyme that we will study that produces this effect. Now, the enzymes I'm going to tell you to know are the ones that are causative of disease and the ones that are limiting a step on each pathway. Okay? So one of these enzymes, which is the step of converting the cholesterol to pregnenolone, right, is cytochrome P450. This cytochrome is found in the mitochondria, okay? And basically, it cleaves the cholesterol side chain, right? Enzyme in position 22, 20, and 22. So this enzyme is a desmolase, right? And it's called cytochrome P450, or the new nomenclature is CYP11A. So the CYP11A is the enzyme that removes carbon groups, right, and converts the cholesterol into the pregnenolone. And this is the happens in the inner mitochondrial membrane, okay? Not in the cytoplasm, but in the mitochondria. So this identification of cytochrome enzymes, okay? There are several P450 cytochrome oxidases that are found in the different areas of the adrenal gland, okay, in the cortex. This here, the table, they were identified, these genes were identified and cloned on the 80s, and obviously they were called cytochromes, all from the same family, P450 oxidase. And the enzymes were named as here. We have the name in there, which is the common name, side chain cleaving enzyme 2022 desmolase. This is telling you the part where the cholesterol is modified and cut. Then it tells you 11 beta hydroxylase. So what does hydroxylase do? Add the hydroxyl group in the 11th part of the carbon beta. 18 hydroxylase, which converts to aldosterone. 17 alpha hydroxylase. 1720 1720 desmolase. It tells you the function. Now you, as I said, you don't need to know exactly this. Also because these names of the genes of these enzymes have changed. Then aromatase and then 20 alpha hydroxylase. Now all these enzymes, P450, have different subtypes, but the nomenclature that is used now is CYP11A1, CYP11B1, CYP11B2, CYP17A1, CYP19A1, and CYP21A2. And each of these enzymes will be expressed in one area of the adrenal medulla. Let's see if I can show you a video, or let me see here in the talk, image, right? I would like to move this slide, okay, into this part here. Because it makes more sense now to show you. So, as I said, there are all these enzymes, CYP11A, CYP11B, CYP11B2, so on and so forth. Each one of these enzymes will affect the cholesterol, but it's localized in an area of the adrenal medulla. So if we look, for example, at a cross section of the adrenal medulla and we stain with antibodies, right, of CYP11B2 aldosterone synthase, can you see? They are found in the zona glomerulosa, these positive cells, okay? If we look at CYP11-1B alpha 11 hydroxylase, it's found in the zona reticularis, okay? And then you can see CYP11B2 in red in the cortex here, well, of the adrenal, and CYP11-1B. You can see also the expression. So each of these enzymes will be identified or confined in different areas of the cortex. That's why it's important to know the cortex areas, because each cortex area will form some enzymes that will catalyze the conversion of cholesterol into different steroid hormones. So let's look at the cholesterol sources for steroidogenesis and the process of what is needed to actually form the cholesterol modification. So the cholesterol is in the blood and has a receptor, and it's internalized, as we show, by calretitin and forming this vesicle that you've seen in the video. Then the cholesterol is released as free cholesterol, and this cholesterol, right, will go into the mitochondria using a molecule which is called transporter or translocator protein, which is called here TSPO. TSPO will act in the outer layer of the mitochondria to internalize the cholesterol, and inside of the mitochondria, inside of internal mitochondrial layer, the enzyme P450 will produce the modification of cholesterol. Okay? So here we have an schematic of the different components like liposomal, cholesterol, scavenge receptor, and the different parts that will modify this initial cholesterol to form the modification. Now, cholesterol cannot travel, because it's hydrophobic, traverse aqueous space between mitochondria and membrane, right? And needs help. So here we have a very simplified mechanism of the represents the layers of the mitochondria. Outer mitochondrial layer and inner mitochondrial layer. Now, for the cholesterol to pass this needs short half-life proteins, and they are called steroidogenesis acute regulator star and the translocator protein, or TSPO. Okay? Also TSPO, formerly known as peripheral benzodiazepine receptor. And how does it work? Into a state resting, right? In the resting state, TSPO binds and colocalizes with the BDAC protein. They make a complex, and cholesterol enters into the inner membrane of the mitochondria, and there is the modification of cholesterol. Also, they can be stimulated by a star and stimulates STPO and BDAC. And what is that through BDAC is that cholesterol then can travel outside of the outer member of the membrane. So obviously this is a cholesterol then can enter out and out of the internal membrane, and then obviously cholesterol is then modified by enzyme. The proper interaction between TSPO and BDAC is not fully understood yet, and there are a lot of studies, right? But it requires this start protein to basically start the mechanism for the cholesterol to enter in the mitochondria and then be modified by the enzymes. So if we look into the steroid synthesis and role of this protein star, basically we have the cholesterol, we have the outer mitochondrial layer and inner mitochondrial layer. So what it does is that cholesterol enters the cell again through the receptors that we just said, TSPO, and the function of a star. It goes into the inner mitochondrial cell layer as cholesterol side chain cleavage enzyme, aka 2022 desmolase. Here, right, the cholesterol will become pregnenolone, and this is the substrate for all the subsequent modifications of the different steroid hormones which we will study, right? And this will start getting complicated, my friends. Okay, I know it's overwhelming, right? But we're gonna go one by one, and also we're gonna study the different functions of it, okay? And basically what we need to know is that each conversion of each different steroid hormone, like mineralocorticoids, glucocorticoids, and androgen, each produced in different zones of the adrenal cortex. So the zona glomerulosa does this pathway, which we will cover after the break. Zona fasciculata does this pathway that produces the cortisol and cortisone. And zona reticularis, which will drive this enzymatic cascade to form testosterone, estradiol, estrogen, and the sexual androgens. So I think we're gonna have a break here, okay? And then we're gonna study one of each of these biochemical pathways to produce each single mineralocorticoid and glucocorticoid. But we will do this after the break, okay? Thank you very much, and I'll see you in 30 minutes. Bye bye, guys. 38